Hello, everyone. Our today's seminar is on the endocrine system. First glands which we are going to be talking about are the adrenal glands. Adrenal glands are a parenchymatous organ. That means they have a stroma and a parenchyma. The stroma is made up of reticular connective tissue and also collagen fibers type 3. The parenchyma consists of the cortex and the medulla. One important thing to note here is that the cortex and the medulla, both of them have different origins. The cortex is of mesodermal origin, and we'll talk about the medulla later on. The cortex occupies most of the slide. When you look at the slide, you can clearly distinguish between the cortex and the medulla, and it is divided into three zones. The first zone is the zona glomerulosa. Uh, it is a very small zone, and it has got these distinct round circular shaped cells, and they are located like right below the capsule, and you can easily distinguish them from any other zones. These cells, they produce these type of hormones which are called as mineralocorticoids, especially aldosterone. The function of aldosterone is maintaining the water and mineral homeostasis, and it does so by reabsorption of the sodium ions from the water. The next zone is the zona fasciculata. This zone occupies most of the cortex, and this zone has the cells which are kind of run parallelly, and they you can see them as fascicles. Uh, they are composed of these special type of cells which are called as spongiocytes, and their cytoplasm contains a lot of lipid droplets, which makes their appearance really light. So you can clearly see the zona fasciculata because all of their cells are really pale. And the spongiocytes, they secrete these hormones which are called as glucocorticoids, especially like cortisol and corticosterone. So these hormones are in charge of regulating the sugar metabolism, and they also help in immunological activities. The last zone of the cortex is your zona reticularis. These cells are kind of like entangled together, and they form a mesh or a network. Their cytoplasm is really dark, and it contains this pigment which is called as lipofuscin. So lipofuscin is just this brownish pigment which you can see in cells which are really old, and it happens due to wear and tear, and it's nothing really dangerous, but it's like a sign of aging. uh, the cells of the zona reticularis, they mainly produce sex hormones, androgens, and dehydroepiandrosterone. So this hormone is like what do you say, a precursor to testosterone. So later on, when it goes into tissues, it gets converted into testosterone. The regulation of the entire hormone system of the cortex is controlled by ACTH. This is a type of a hormone which is produced in the pituitary gland. All hormones which are produced in the adrenal cortex are steroid hormones. So what this basically means is that there are hormones basically have three origins. They can either be from steroids, amino acids, or peptides. So all the hormones which are produced in the adrenal cortex are steroid hormones. Next, we have the medulla of the adrenal gland. It is derived from the neuroectoderm of the neural crest. So this is different from the mesodermal origin of the cortex. And the medulla, the cells over here, they are really dark, and the endocrine activity of them is regulated by chromaffin cells and ganglionic cells. Chromaffin cells regulate most of the endocrine activity, and there are two types of chromaffin cells. Most of them, almost 80%, they produce epinephrine or adrenaline, and a few of them, approximately like 20%, they release norepinephrine or noradrenaline. These chromaffin cells, they have a different origin because they are from the neuroectoderm. So basically, they arise from the sympathetic postganglionic neuron. Basically, when this postganglionic neuron of the sympathetic origin loses its dendrites and axons, it results in the formation of a chromaffin cell. Chromaffin cells also act as transductors. So what this means is that basically they convert an electrical signal which they get from the ganglions and the neurons into hormones. And the reason why they are transductors is because they kind of have a connection with the preganglionic nerve fibers. One last thing, epinephrine and norepinephrine are derived from the amino acid tyrosine. So they are of amino acid origin, unlike the hormones from the cortex, which are of steroid origin. So now I will just show you guys the slide. Okay, so this is a slide of the adrenal gland. You can see that it is surrounded by adipose tissue. So this could be one of the features that helps you identify the slide if you get it in your slide test. So when you zoom in, you can see the connective tissue capsule over here. And when you zoom in a little bit more, you can see that this is the cortex. And this purple area over here inside, all of this is the medulla. It's really dark, like I had said before. And everything else in the slide here, all of this stuff is just your cortex. So let's look at it in a bit more detail. Yeah, so right below the dense connective tissue capsule, the first layer is going to be a zona glomerulosa. So over here, you can see that these cells, they are like a bit circular and they are not long or anything. And you can see there's barely a layer or two of these cells over here. Like see, for example, this one, this one, this one. And next we have the zona fasciculata. You can immediately see a change over here. Like these are like longer strands over here. Even this, these are really, really long strands which run parallelly to each other. So most of this part is your zona fasciculata, and the last layer is the zona reticularis. You can immediately recognize this because of the amount of lipofuscin which is present over here. And also these cells are just arranged really randomly because they form a network. And apart from that, lipofuscin is a clear indication of this zone. Then we can clearly distinguish between the cortex and the medulla. There's a clear distinction boundary over here. And if you zoom in inside, all of these cells which you can see are chromaffin cells. There'll be really few ganglionic cells here and there, but they're pretty difficult to spot. And the chromaffin cells, there are of two types, right? Some of them secrete epinephrine, others secrete norepinephrine. But in our microscopy, you can't really differentiate between them. You can only differentiate between them in electron microscopy. So yeah, this is it. This is pretty much what your adrenal gland looks like, and I hope it was okay to understand. Moving on to our next topic, the endocrine function of the kidney. So the kidney also releases a few hormones which are really important. The first one is a hormone called as renin, and it helps increasing our blood pressure. Uh, the way it works is that it splits this uh, compound called as angiotensinogen, which is produced in the liver, and the renin like acts on it and splits it to form angiotensin 1. This then gets converted into angiotensin 2 in the lungs, and angiotensin 2 is basically a vasoconstrictor. That means that it increases the blood pressure. Next, we have the erythropoietin. It is produced in the renal cortex and it acts on the stem cells of the bone marrow to basically increase erythropoiesis. Erythropoiesis is just the formation of red blood cells in the bone marrow. Next, we have the vitamin D, and the kidney basically helps in like activation of vitamin D from its precursor to its actual active form. And in the end, we have prostaglandins. So these few are the examples over here. Yeah, these few are the examples over here which you can see of prostaglandins. You really need to remember their names, and they basically help in maintaining the blood flow and the glomerular filtration rate, which is basically the rate at which urine is filtered. Next, we have the juxtaglomerular apparatus. So these are a bunch of cells which interconnect, and they are really important in a bunch of physiological functions. There's three types, yeah, juxtaglomerular cells, microdensa, and the extragranular mesangial cells. Now, unfortunately, the slide correlates a lot with your urinary system, which you haven't done yet. So I will just quickly give you a brief over what you need to know for this part to understand this better. So, juxtaglomerular cells are found near the afferent arteriole. Okay, so this is your afferent arteriole. This is where like the blood vessels enter into your nephron. So this slide is basically of the nephron, this picture over here. And yeah, this is the afferent arteriole. So this is where your juxtaglomerular cells will be found. Next, we have the macula densa, and It is found near the dense convoluted tubule. So this is a dense convoluted tubule. And last, we have the mesangial cells. These are found in the Malpighian corpuscle. Malpighian corpuscle is nothing but glomerulus and Bowman's capsule together. So this entire structure, which you can see here, this is going to be our Malpighian corpuscle. So this is the overview which you need to know to understand what I'm going to be talking about now. The juxtaglomerular cells, like I said, they're located in the wall of the afferent arteriole, and their main function is also they act as transductors. The cytoplasm of these cells have secretory granules which release renin, and I already spoke about renin before. Uh, but renin is released under three circumstances. The first circumstance is the adrenergic stimulation. This basically means that when the sympathetic nervous system is activated, this can be during stress or during low blood pressure, as far renin is released, due to which the blood pressure can be brought up high again. The next condition is if there is a decrease in renal perfusion pressure. What this means, basically in simple terms, is when blood pressure drops. This can be due to dehydration or blood loss or other factors like that. And the afferent arteriole here detects that the pressure is reduced of the blood, and this then stimulates the renin release. The last case when renin is released by the juxtaglomerular cells is when there is a decrease in the NaCl absorption in the macula densa. So I'll talk about the macula densa shortly, but basically if the NaCl levels are really, really low, this can be due to low pressure or reduced filtration rate or any something like that. Then the macula densa signals the juxtaglomerular cells to release renin. Okay, moving on to the macula densa. It's just a few group of cells which is located in the DCT, and the cells over here they become columnar, and the apical surfaces of these cells come in contact with the primary urine. These cells detect the urine flow and the NSL concentrations, like I just said, and they in turn communicate with the juxtaglomerular cells in case the blood pressure needs to be brought back up. Last, we have the extragranular mesangial cells. These cells are just a connection between the juxtaglomerular cells and the macula densa, and they are located in the Malpighian corpuscle. Lastly, we have the epiphysis. This is also of neuroectodermal origin, and it has connective tissue septa, which divide the gland into incomplete lobules. The cells which have the endocrine activity in the epiphysis are called as pinealocytes, and they also function as a transductor. So there are three cells in your entire endocrine system which function as a transductor: pinealocytes, the juxtaglomerular cells, and your chromaffin cells from the adrenal gland. Okay, these three are the examples which you have to know for what function as a transductor. They receive their innervation from the autonomic nerve fibers, unlike in juxtaglomerular where they receive from sympathetic nerve fibers. But yeah, the pinealocytes are star-shaped, and they are endings of the star-shaped processes. They have secretory granules which contain the hormone called as melatonin. This melatonin, it acts as a modulator for multiple endocrine activities, and it reaches its peak in the nights, and it is really, really low in the mornings. So this technically functions like an internal clock. The melatonin, it kind of appears from serotonin. So when serotonin undergoes a bunch of reactions and it gets transformed into melatonin. Melatonin is necessary for us to fall asleep, and when we're asleep, it also helps in the reparation of of the organism. It also has anti-aging properties, and it also signals the glands to suppress the activity of sex hormones. Next, we have something called as the astroglial cells. They are just a modification of the astroglia, nothing much to be spoken about for this. One important thing is that approximately after 7 years of life, the epiphysis undergoes like some sort of regression, and a bunch of these calcified structures, which are called as acervolos cerebri, appear in the parenchyma, and they just keep on increasing as we grow older. So this is it for me.
All right, guys. So now we'll be talking about the hypophysis or the pituitary gland. So this is the slide of the pituitary gland, and this is the anatomical diagram telling you about the parts. So first of all, with the pituitary gland, it's divided into two lobes. You have the anterior lobe and then you have the posterior lobe. The anterior lobe is also called the adenohypophysis, which is this part, and the posterior part is called the neurohypophysis. Okay. So the anterior lobe, it has three parts. These are called pars tuberalis, pars distalis, and pars intermedialis. And the posterior lobe, it has three parts as well. These include the median eminence, infundibulum, as well as the nervous lobe. So now, when we talk about the anatomical position of this pituitary gland, it is located under the third cerebral ventricle, and it is connected to the thalamus, hypothalamus, as you can see here, via the infundibulum, which is this part. Sorry, and it is near the optic chiasma. And when we talk about the anterior lobe, it is mainly responsible for hormone secretion, while the posterior lobe it's involved in storing and release of the neurohormones from the hypothalamus. So when we talk about the origin of the hypophyses, the adenohypophysis is derived from ectoderm, which is also called Rathke's pouch, and it is made up of numerous epithelial endocrine cells, which are arranged in anastomosing cords. When we talk about the neurohypophysis, it is originated from neuroectoderm, and it consists of unmyelinated nerve fibers and pituicytes. So when we talk about ectoderm and neuroectoderm, you will learn more about this in embryology, and we will explain you during that. But these are just layers through which an embryo forms. These are the initial layers. These include ectoderm, endoderm, as well as neuroectoderm. There's mesoderm as well. So these are just different layers from which different organs essentially develop. Now, now we're looking at the different parts of the slice, the adenohypophysis and the neurohypophysis. So as I said before, this is the adenohypophysis and this is the neurohypophysis. Now, when we talk about the pars distalis of the adenohypophysis, right, it is made up of two types of cells. You have the chromophobes and then you have the chromophils. When we talk about the chromophobes, as you can see here, this picture is from your lecture. When we talk about the chromophobes, they are undifferentiated cells and they are degranulated chromophil cells which have a stellate shape. When we talk about chromophils, they're divided into two types of cells. These include, acidophils, these ones, and then basophils. When we talk about the acidophils, they are involved in production of prolactin as well as growth hormone. When we talk about basophilic cells, they are involved in production of follicle stimulating hormone, LH, TSH, thyroid stimulating hormone, or ACTH. Here also on this slide you can see chromophobes. So now you can see the difference between them. When we're talking about chromophobes, they have a lighter appearance. When we talk about the acidophils, they have, a more pinkish color appearance. And then when we talk about basophils, they are, more darker compared to, acidophils as well as chromophobes. When we talk about the pars tuberalis of the adenohypophysis, it produces FSH as well as LH. When we talk about the pars intermedialis, which is here, which is the transition between, the adenohypophysis, this part is just, this is zoomed in version of this part. So pars intermedialis is the part between the adenohypophysis and the neurohypophysis, and it is important in production of the MSH, which is the melanocyte stimulating hormone. Now, moving on to the basophilic cells specifically, they stimulate the secretory activity of the peripheral endocrine cells, and they have different types of cells. These include adrenocorticotropic cells, which stimulate the adrenal cortex to produce cortisol. Then we have gonadotropic cells, which include follicle stimulating hormone, which stimulates the ovarian follicles to release estrogen. And then you have the luteinizing hormone, which stimulates the corpus luteum to produce the progesterone. And then you have ICSH, which stimulates the Leydig cells, which results in androgen production in males. And then the third one is the thyrotropic cells, which releases TSH, which stimulates the thyroid gland and results in increase in the level of T3 and T4. So we have three types of basophilic cells. These include adrenocorticotropic cells, gonadotropic cells, as well as thyrotropic cells. When we talk about acidophils are divided into two. No, when we talk about chromophils, they're divided into two types of cells. We have acidophilic cells and basophilic cells. So don't confuse that. When we talk about the acidophilic cells, right, they're divided into two types of cells as well. They're called somatotrophs as well as mammotrophs. When we talk about somatotrophs, these release the growth hormone, which is important in, stimulating the liver to produce somatomedins, which promotes the growth plate expansion in the liver. When we talk about the mammotrophs, they produce prolactin, which is essential for the lactation, and this hormone levels basically increases during pregnancy. So just to summarize, we have adenohypophysis, neurohypophysis. Adenohypophysis is made up of chromophils, which are mature cells, which are divided into acidophils, basophils. Then you have chromophobes, which are undifferentiated cells, which are very light and have a stellate shape and do not have much function except some metabolic function. Acidophilic cells include prolactin as well as growth hormone. And when we talk about basophilic cells, these include gonadotropic cells as well as adrenocorticotropic cells as well as thyriotropic cells. Now, when we look at here, these two are part of the neurohypophysis. These include pituitary and then you have the fenestrated capillaries. This is not important. This is just one of the characteristics which is seen on the slide. You know how it is a neurohypophysis because here you can see, the red blood cells. Now, when we talk about the blood supply of the hypophyses, the atrial supply, which is the blood flowing in, it is done by the superior hypophyseal artery as well as the inferior hypophyseal artery. So it is basically in their name, the superior one supplies the blood to the upper part, including the anterior pituitary, and you have the inferior hypophyseal artery, which provides the blood to the lower part. When we talk about the hypothalamo-hypophyseal portal system, this is made up of a primary capillary plexus, which is present in the median eminence of the hypothalamus. And what it does is it is involved in collecting and releasing the hormones. Okay, these hormones are essentially transported through the portal veins to the secondary capillary plexus in the anterior pituitary, where they regulate hormone secretion. When we talk about the blood circulation in the posterior pituitary, it is important to remember that it does not produce hormones, but it is involved in storing and releasing of the hormone, which mainly include oxytocin as well as ADH. When we talk about the movement or the transport of these hormones enter the capillary plexus of the neurohypophyses, where after that they're released into the bloodstream. When we talk about the venous drainage, which is the blood flowing out, the blood drains through the hypophyseal veins, then into the cavernous sinus, which ensures the proper circulation of the hormones throughout the body. So just to summarize, the atrial supply, inferior hypophyseal artery, and the superior hypophyseal artery, again for the upper and the lower part of the pituitary gland. Then you have the hypothalamo-hypophyseal portal system made up of two capillary plexuses, and then again, I repeat that the neurohypophysis, it is not involved in the production of the hormones. It is involved in releasing the two hormones, oxytocin and ADH. And the venous drainage is done by the hypophyseal veins into the cavernous sinus. When we talk about the hypothalamo-hypophyseal-neurosecretory system, so this basically means is that the hypothalamus, it basically regulates the activity of the pituitary gland by releasing special hormones known as the hypophysiotropic regulatory hormones. So these are divided into two categories. We have liberants and we have statins. When we talk about liberants, these are releasing hormones and they stimulate the pituitary gland to release specific hormones. So we have corticotropin releasing hormone, gonadotropin releasing hormone, thyrotropin releasing hormone, and somatotropin releasing hormone, and they all have different functions such as CRH. It triggers the release of ACTH. When we have gonadotropic releasing hormone, it stimulates the release of FSH as well as LH. And when we have thyrotropic releasing hormone, it promotes TSH as well as prolactin release. When we talk about statins, these are inhibiting hormones. They suppress the release of certain pituitary hormones. These include somatostatin, which inhibits the secretion of growth hormone. And then when we talk about prolactin inhibiting hormone, it prevents the prolactin release. So the main function of the hypothalamus is to, homeostasis, basically maintain the hormone balance as well as controlling metabolism. So again, hypothalamus, it regulates the activity of pituitary gland. For example, it will release liberants, which will act on the pituitary to cause it to release specific hormones, or statins, which suppress or stop the activity of the pituitary gland. Now, when we talk about specifically neurohypophyses, it has a neurosecretory system which is made up of neurosecretory cells, which are specialized neurons, and they're responsible for hormone synthesis and release. Okay, so when we talk about specific characteristics of these neurons, they have cell bodies in the hypothalamus, and they have a supraoptic nucleus as well as paraventricular nucleus. Okay, and they send their axons down to the posterior pituitary. And when we talk about their axons, yeah, they contain Hering's bodies, which are accumulations of secretory granules that store hormones. And this is the picture from the slide itself, which was here. If you zoom in into this part, you can see Hering's body somewhere here. When we talk about the different hormones which are stored in the neurohypophyses, as I said before, these include vasopressin, also called ADH, as well as oxytocin. When we talk about vasopressin, it is involved in regulating the water balance by controlling the kidney function, and it helps constrict the blood vessels to maintain blood pressure. When we talk about oxytocin, it is involved in stimulating uterine contractions during childbirth as well as promoting milk ejection. Another thing I forgot to mention is that the hormones which are released by the neurosecretory system into the bloodstream, this is done by the process of exocytosis. Now, lastly, when we talk about the diffuse neuroendocrine system, this is not really important, but if you know 2, 3 points about it, it's okay. So when we talk about the diffuse neuroendocrine system, it consists of scattered endocrine cells, which are found in small groups within different tissues or along non-endocrine cells as well. So the main features of these include that these endocrine cells, they store and release hormones. They produce very low molecular weight polypeptides. They were initially discovered in the gastrointestinal tract, and then later they were found in the central nervous system. They work with the neurotransmitters to also regulate the synaptic activity, as well as their signaling can be done through endocrine, which is through the bloodstream, paracrine, which is affecting the where a cell basically releases a particular secretion, and this affects the surrounding cells, or neurocrine signaling, which is just acting like neurotransmitters. When we talk about where they are found, they're found in various locations throughout the body. These include the gastrointestinal tract, urinary tract, reproductive system, respiratory system, parafollicular cells of the thyroid gland, chromaffin cells in the adrenal medulla, various locations. Okay. So the main function of the diffuse neuroendocrine system is that it is involved in the hormone regulation through different organs, as well as it helps maintain body functions such as, digestion, metabolism, stress response. So that was it.
Okay, hi guys. So today we're going to talk about the thyroid, the parathyroid, and the pancreas. So in terms of theory, it's actually very simple. I'm just gonna go over the basic morphological features with you guys, show you guys how it looks like in the slide, and then the more in detail theory, you guys are obviously just gonna have to go through it on your own for the sake of time. So starting with the thyroid on the surface is going to be covered by dense connective tissue from which the septa is going to emerge, and the septa is actually going to divide the parenchyma of the thyroid into lobules, which you guys will see. Now, the most important feature of the thyroid, guys, the thyroid is a slide that you guys are going to see it from a distance. Like you can pick up the slide in your hand and you will be able to know it's the thyroid just by putting it against the light, because you guys are going to see follicles. And these follicles, like, you're not going to confuse it with anything else. Some students think that the follicles look similar to like follicles in the ovary, but they really do not. This is going to be very specific to the thyroid. As soon as you guys see these follicles, you know you're in the thyroid. And these follicles are going to contain a single layer of follicular cells that are going to be sitting on top of a basement membrane. And the follicles are going to contain colloids. The follicular cells, they are neuroendocrine cells. They secrete. In this case, they're going to secrete T3 and T4, and it's going to be stored in the follicles as colloid. You're also going to see in this slide parafollicular cells, which are going to secrete calcitonin. And here at the bottom, I just included a small diagram that shows the morphological changes with the follicular cells. This is found in the lectures that are available for you guys. You can have quiescent follicles And follicles that are increased in function, that are actively storing, secreting and storing the colloid. And you can see the change in the shape of the follicular cells when you have a quiescent follicle that's much larger. And they kind of have like a, almost one could say a squamous sort of shaped, versus when there's an increased function, you could say they have almost a columnar sort of shape. This is something that is not very easily seen on the slide, but it's good to just note that there is a change in the morphological shape, or in the shape of the follicular cells, based on the function of the thyroid. This is good to know because they might ask you this in theory. Now, just looking quickly at the thyroid. So I'm going to zoom out. So this is what you guys are going to see. And if you hold it against the light, you can instantly see, like, you might not even understand what you're seeing, but you know it's definitely it's going to be definitely different from all your other slides. And if you zoom in, you're going to see these like circles that seem to have this gelatinous amorphous substance. This is the colloid, guys, and it's going to be filled in the entire slide. The thyroid is just basically filled with these circles that seem to have this amorphous substance. And these are the follicles, and this is colloid. And if I zoom in to any, I can see the layer of follicular cells, as is very similar in the diagram that I just showed you guys. Here's the colloid, here's the follicular cells, and all the cells in between are going to be these cells in between are going to be your parafollicular cells. And so this is the thyroid slide. It's very, very simple. There's really not much to it. You guys are just going to see colloid. As soon as you guys see these follicles with colloid, you know that you are in the thyroid. There's really not much to it. Um, Now, going back to our parathyroid is also encapsulated by dense connective tissue, and the septa is also dividing the parenchyma into lobules. And you're going to see cells that are either going to be arranged in strands or in groups. Personally, I think for these for our histology slides, they're mostly arranged in groups, but we'll see. And there are two main cells that you see in the parathyroid. You have your chief cells, which you guys are going to very obviously easily see. They have a light cytoplasm because they contain a lot of glycogen, and they secrete the hormone parity hormone. They contain secretory granules. Then you have your oxyphil cells. These are larger, more eosinophilic, and they're more eosinophilic because they are abundant in mitochondria. Now, oxyphil cells have no secretory granules, so they're not going to be releasing anything, and it is believed that they are a degeneration of the chief cells. Now, to be completely honest with you guys, our slides for parathyroid, from my memory, looked different from the ones I saw that you guys have, and it was a little bit difficult, like, to kind of be able to properly show you guys what the oxyphil cells would look like. So I found some images on Google. Like, you guys definitely see it on your slide, but it's better for you guys to see that image first, which shows specifically what it looks like, and then you guys will easily be able to find it in your slides. So this is the parathyroid. Again, just zooming out, and as soon as I zoom in, I'm seeing like two different sorts of cells here. I'm seeing these pretty dark, small, circular, round cells, which these are going to be your chief cells, as you guys can see. And then I struggled a bit to find an oxyphil cell. I would guess like this would be an oxyphil cell. However, for your slide test, if you guys are struggling, obviously you guys are going to recognize that this is going to be the parathyroid, because you're literally just, this is all you're going to see. Just these oxyphil and chief cells. You're not going to see much else, and obviously vessels and stuff, but you're not going to see much else. You're not going to see any colloid. You're not going to see islets of Langerhans. And you guys are just going to have to do process of elimination and know that this is the parathyroid. And so, yeah, this is pretty much all you're going to see, and you guys are going to talk about in theory. And you know that if the professor points to a cell that you know is very obviously not this dark thing, then he's pointing to an oxyphil cell. And so I found this image that could kind of better try to show you guys what I'm talking about. So you have the chief cells, which are small and dark, and then you have these larger like cells. This would be like the oxyphil cells that have a more eosinophilic like cytoplasm to it. Uh, right. So now we're going to go into the pancreas. The pancreas is a good slide. It's also very, very easy to recognize. It's going to contain structures that no other slide contains. But for theory wise, just know the pancreas is a parenchymatous organ. It contains two parts. This is very important. Like this is like I won't say the most important thing, but you guys need to know this. The pancreas contains the exocrine part and the endocrine part. Exocrine and endocrine. Do not forget this. And do not forget that the exocrine part is composed of two things. It's composed of serous acini and the ducts. A lot of students tend to forget this and they just say, oh, the uh, exocrine part of the pancreas is just the acini, and it's not, guys, because how's the acini going to travel? Where's it going to travel through? It's going to travel through the ducts. Um, you guys know that the pancreas, or like obviously I can't ask you this, but the two main functions of the pancreas, yes, it's going to secrete insulin. This is the endocrine part of the pancreas. The pancreas also releases digestive juices, and it does this to the serous acini, and that's what's going to be traveling through the ducts. So when we talk about the exocrine part, we're talking about the serous acini, which is going to kind of make that digestive juice, and it's going to travel through the ducts. So you need to include the ducts. And another way the professor might ask you, how else would you refer to the pancreas? The pancreas is a compound alveolar, pure serous gland with islets of Langerhans. So again, the pancreas is a parenchymatous organ that contains its exocrine part. The exocrine part makes the digestive juices. The juices need to travel through the ducts. And then you have the endocrine part, and the endocrine part is composed of the islets of Langerhans, which are going to, most importantly, make insulin. And then, yes, it's a compound alveolar pure serous gland with islets of Langerhans. It is very important. So this is a basic overall picture of the pancreas. Obviously, I will go more in detail and show you guys what it looks like in Fingerland. But just know that the two main things that you guys are going to see from a distance, I believe the pancreas is also one of those slides where I knew that if I held it against the light and I saw like dark background, dark, very purplish background, and then these circular light white parts, I knew that 9 times out of 10 I was looking at the pancreas, because this is going to be the serous acini, and then these white parts are going to be the islets of Langerhans, and then a bunch of ducts, and then you're going to see a bunch of ducts, and you know that you're in the pancreas. Now the ductal system, I'm not gonna go over this because this is something that you guys are going to have to go over multiple times. It's very important. And if you guys need help, if you guys are struggling to understand the ductal system, like please reach out to me or anyone else because you guys need to understand this. I just included this image very quickly. So you're going to have your serous acini, which is going to produce the juice, and the juice is going to travel through your intercalated duct, your intralobular duct, because remember it's going to be divided into lobules, and then you're going to have your interlobular, as in lobules that are now outside, into your interlobar duct, which is the bigger ducts in going into your main duct. It sounds confusing. There are so many different images that you could find in Google to try and understand the ductile system. Think of it like a tree branch, guys. Okay, you have the leaf that's going to be attached to those tiny little branches. Then you have you have the main branch, which will have some smaller branches, and those smaller branches will have even smaller branches, which are going to be tied to the leaves. This is what this is. You have the big branch, then you're gonna have some middle-sized branches, and each middle-sized branch is gonna have some smaller branches, yada yada yada, and it's just going to kind of branch out. You need to know they will ask you about the ductal system if you get the pancreas, as if they will also ask you that if you get any other gland. But just know that if you get the pancreas, they will also 100% ask you this. And once again, exocrine part will be the dark stained part, which will make up most of the parenchyma, composed of the acini and the ducts. And you have the endocrine part, which is the islets of Langerhans. So the exocrine part, going to a bit more detail, is going to contain two types of cells. You have your acinar cells and your central acinar cells. Your acinar cells are going to make up 80% of the pancreas. and they are going to produce your digestive enzymes such as amylase, elastase, which are going to be produced in like its active form, and then you have trypsinogen, which is in its inactive form. And this is important because it could be, I guess, the right word would be kind of volatile and harm the pancreas, which is why it needs to be secreted in its inactive form. And later on, it's going to be activated when it's needed. So you also will secrete a trypsin inhibitor, again to prevent the activation of this proteolytic enzyme while it's still like within the acinar lumen and the ducts, to avoid it from damaging the cells and the ducts. And these the they're going to be stored in zymogenic secretory granules. And just know, it's important for you guys to know that exocytosis is going to be stimulated by CCK. You then have your central acinar cells, and they're going to produce this alkaline secretion, which is composed of water and bicarbonate. This is a very important component of the pancreatic juice. And know that this is going to be stimulated by secretin. Another important thing for you guys to know, all of the epithelial cells in the acini and the ducts are going to be joined by tight junctions. And this is because, again, the pancreas is going to be producing and secreting these very like volatile digestive enzymes, and they're proteolytic. They're just gonna like destroy what they're surrounding. So it's important that they are within like acini and ducts that are joined by tight junctions to prevent them from leaking and then like damaging the ducts and the acini. Uh, and you also have secretion of protective substances, again the trypsin inhibitor and bicarbonate ions. And this is just an example. So this, all of this dark thing here, all of this is the exocrine portion. And then you guys know by now how ducts look like. And here you have your ducts. Um, and these are just some more images of how some ducts look like. Again, for sake of time, I'm not gonna go in detail about this. Just try your best to like study and try to differentiate like how an interlobular versus an interlobar duct looks like. And every time you guys are looking at ducts, try to imagine a tree branch, because this is what it is. It's all connected as if it were a tree branch. And then the endocrine function, the islets of Langerhans, you have more than 1 million islets in the human pancreas. They're mostly abundant in the tail. They are polyhedral or rounded cells. They're arranged in cords. They're separated by sinusoidal capillaries. And their secretory activity is going to be regulated by autonomic nerve fibers. Sorry for this mistake here. And there are three main cells. You have A, B, and D cells. The A cells are larger, but they're smaller in number, and they secrete glucagon. The B cells are the most numerous. They make up 70% of the cells, and they're going to secrete insulin. And then you have D cells, which are smaller, which are going to secrete somatostatin. Um, Now, just quickly going to show you guys, we have two pancreas slides. So I'm going to zoom out. This is how it looks like. Zooming in, you guys, and this is what I mean by if you put it against the light, you can see some very dark like areas, and then you see these very light areas. Again, if I zoom in, I'm seeing just a lot of like dark celophilic acini. You guys know how acini looks like. And then randomly scattered within the acini, you're going to see like cords of cells that are much lighter than the acini. And this is an islet of Langerhans. This is the islet of Langerhans, which is going to contain those three types of cells secreting glucagon, insulin, and somatostatin. Again, so I'm moving across the pancreas. Again, acini for digestive juice, and then I stop. Oh, this is an islet of Langerhans. As I move across, you're not going to move much without finding an islet of Langerhans. With every skull that you do, you're going to find an islet of Langerhans. They're just going to be the cells that are very different from the acini, and they're arranged in like cords or groups. And then, of course, you guys are also going to have your ducts scattered around. And this is it for the pancreas. This is the other pancreatic slide that you guys have. Again, you could easily notice the difference in color or shade between the acini and the islets of Langerhans. Uh, this is very, very different from the thyroid. There's just no way for you guys to confuse this. There's no follicles. You don't see colloid. You don't see that amorphous substance. You see two different shades of cells, a bunch of ducts. You are 100% in the pancreas. Thank you guys.